Good day to the viewers. Today we are going to discuss about RNA and its types. Now let's see the introduction of ribonucleic acid. Ribonucleic acid is a single standard nucleic acid molecule and made up of ribonucleotides. A ribose nucleotide in the chain of RNA consists of ribose sugar, phosphate group and a base. In each ribose sugar, one of the four bases is added: adenine, guanine, cytosine and uracil. The base is attached to a ribose sugar through a glycosidic bond, and phosphate connects nucleotides via phosphodiester bond. As RNA comprises many ribose nucleotides, the length of the chains of the nucleotides can vary according to their types or their functions. Properties of RNA. RNA is a single stranded molecule and not a double helix. One of the consequences of this is that RNA can form a variety of three-dimensional molecular complexes than DNA. RNA has ribose sugar in its nucleotide rather than deoxyribose. These two sugars differ from each other in the presence or absence of an oxygen atom. Ribose sugar is also a cylindrical structure consisting of 5 carbon and 1 oxygen, just like DNA. But the major difference in the presence of extra hydroxyl group in second carbon of ribose, which is absent in deoxyribose sugar. The hydroxyl group in second carbon atom makes the RNA molecule prone to hydrolysis. RNA nucleotides carries the nitrogenous bases purines and pyrimidines, but in RNA, in place of thymine, uracil is present, which also forms hydrogen bonding with adenine. Now move on to structure of DNA. RNA is a typical single-stranded biopolymer of ribonucleotides bonded with each other via a phosphodiester bond. An RNA strand is synthesized in the 5' to 3' direction from a locally single-stranded region of DNA. It has ribose sugar that are attached to four bases: adenine, guanine, uracil and cytosine. Ribose sugar has an extra hydroxyl group in second carbon atom. This extra hydroxyl group in RNA has led them to be synthesized for short-term functions. The three-dimensional structure of RNA is critical to its stability and function. RNA, being a single-stranded molecule, can form a complex structure by allowing its ribose sugar and bases to be modified on the action of cellular enzymes to perform different functions. They are even capable of folding on themselves and showing intramolecular hydrogen bonding between complementary strands, making it a double-stranded molecule to exhibit specific functions. Types of RNA. Of many types of RNA, the three well-known and most commonly discussed and found in almost all organisms. They are messenger RNA, ribosomal RNA, and transfer RNA. Messenger RNA. It is a single-stranded RNA molecule that is complementary to one of the strands of DNA. mRNA is the version of genetic material that leave the nucleus and move to the cytoplasm, where responsible proteins are synthesized. This RNA has utmost importance during protein synthesis. When the ribosomes move along this mRNA, it reads the base sequences and uses the genetic code to translate them into specific proteins. These codes are in the form of triplet sequences of nitrogenous bases and are often referred to as codons. As we know that mRNA is responsible for transferring the genetic information into ribosomes, whereby reading the base sequence on mRNA, the translation of protein is made possible. Thus the name resembles its function, that is messenger RNA. Without mRNA, DNA information could not be used. Ribosomal RNA. It is a single-stranded RNA molecule found in cells that forms the part of the protein synthesizing organelle ribosome. It is synthesized inside the nucleus, particularly in the nucleolus where rRNA coding genes are present. The synthesized rRNA can be of varying sizes, commonly distinguished as small and large. These newly synthesized rRNAs combine with ribosomal proteins and form smaller subunits and larger subunits of ribosomes respectively. These rRNAs are vital in recognizing conserved regions of incoming mRNAs and tRNA, thus facilitating their binding and carrying out protein synthesis. Additionally, rRNA also has enzymatic activity, that is peptidyl transferases, and catalyze the formation of peptide bond in between two aligned proteins or amino acids during protein synthesis. Transfer RNA or tRNA. It helps to decode information present in mRNA sequences into specific proteins. It is encoded by DNA in the cell nucleus and transcribed with the help of RNA polymerase III. The structure of tRNA folds upon itself and creates an intra-complementary base pairing which gives rise to hydrogen bonded stems and associated loops that contain nucleotides with modified bases. The structure in two-dimensional resembles a clover leaf having three loops and an open end, which are usually consist of 75 to 90 ribonucleotides in length. Each of these loops consisting of arms has a distinct name and function. The three loop consisting arms are namely D arm, which has recognition site for specific enzyme amino acyl tRNA synthetase. T arm that consists of ribosome recognition site and anticodon arm that recognize and bind to mRNA present in the ribosome. The open end with no loop is the site for attachment of amino acid via 3' hydroxyl bonding with carboxyl group of the amino acid. In general, tRNA reads the code on the mRNA sequence in ribosome and translates specific amino acid. It does so along the length of the mRNA and gives out a polypeptide chain of amino acid in association with other important enzymes like amino acyl tRNA synthetase and peptidyl transferase. Other types of RNA. Ribozymes are otherwise called as catalytic RNA. These types of RNA refer to those RNAs that are capable of showing enzymatic activities. They were first discovered in introns of precursor ribosomal RNA of Tetrahymena thermophilus, where it was found that these non-coding sequences were capable of excising themselves without any protein or external source. These RNAs plays a vital role in major reactions like RNA splicing, viral replication and tRNA biosynthesis. rRNA also shows enzymatic activities and thus can be termed as ribozymes. Example, peptidyl transferase activity in ribosomes. Antisense RNA. Antisense RNA that contain sequences that are complementary to protein coding sequences of mRNA. These are single stranded like mRNA but cannot code for proteins. However, they can interfere and inactivate their complementary mRNA sequences, thus inhibiting protein synthesis. This ability of antisense RNA has led researchers to create artificial antisense RNAs that can inhibit the protein synthesis of potential disease causing organisms or of infected cells, which then end up killing unwanted cells. Functions of RNA. The prime function of RNA is in protein synthesis. Without RNA, the information encoded in DNA could have never been transcribed to make essential proteins that a cell needs to maintain its integrity. mRNA have now been widely used in pharmaceutical industries to synthesize potential vaccines. Moreover, mRNAs are now used to develop new categories of medicines. mRNA have made the formation of the cDNA library possible. rRNAs are structural units of ribosomes which are essential organelles during protein synthesis. Ribozymes can help suppress the expression of specific mRNA by cleaving them out. Artificial antisense RNA are capable of arresting protein synthesis by binding with mRNAs, which have contributed to the human's ability to combat diseases and mutations. Thank you for listening the video.